Good afternoon, and welcome to our weekly Research in Action, brought to you by the Division of Research at Florida Atlantic University. My name is Karen Scapinato. I will be today's moderator. It is my great pleasure to introduce today's speaker. Dr. Krista McCoy is one of our most recent hires here at FAU. She received her Ph.D. in interdisciplinary ecology from the University of Florida and has broad training in ecological and developmental toxicology, physiology, and behavior. Her postdoc fellowship was supported by NIH, and then she moved to East Carolina University, where she earned tenure. And she then, this summer, joined our Harbor Branch Oceanographic Institute as an associate research professor. Her current research focuses on understanding how we can prevent common disorders caused by environmental pollutants, including harmful algal toxins. With that, I'm going to turn it over to Dr. McCoy, who can tell us more about how to protect people from toxicants.
Hi, good morning. I hope everyone can hear me. And if you cannot see my screen, somebody scream out and let me know. Today, I want to tell you a few stories about how I have come to study the ways in which we can protect people from toxicants in the environment. Okay, so the first thing you should know is that there are tens of thousands of synthetic chemicals in use currently. And only about 2% of those have been evaluated for human health effects. So the vast majority have not been studied. Just over half of those chemicals that have been evaluated are known to disrupt the endocrine system. And so the endocrine system is the system that makes our hormones and regulates our body functions. So it controls our physiology, and it spans the many organs, almost all of our organs, from the brain, the thyroid gland, the thalamus, or thymus, and our pancreas, adrenal glands, that's where our stress hormones are produced. And also our gonads, so ovaries in women that produce eggs, and the testes in men that produce sperm. And one thing I want to draw your attention to is that a single chemical can alter many different organs. And so organotins are chemicals that are found in paint and many different plastics, PVC and vinyl, and we're exposed to them sort of all the time because they're in so many different items that we use. And we know that organotins can affect the pancreas, which is an endocrine organ. And through these specific mechanisms, we know that organotins can influence insulin and lead to impaired glucose homeostasis, or the way and the concentration of glucose in our body. And that is associated with diabetes. We also know that these chemicals can affect our central nervous system, and people have studied the ways in which they affect our brain. And this is really interesting because these chemicals affect our hunger center, and so they make us hungrier. And this leads to obesity. Other studies have found that organotins actually target fat tissue specifically, so our adipose tissue, and they've studied the specific mechanisms of toxicity that lead to this differentiation of pre-adipocytes, so pre-fat cells, into adipocytes, the cells that make up our fat. And this also leads to obesity. Other studies have shown that organotins can reprogram cells to become fat instead of bone cells. So certain cells are, during development, are destined to become different things. And organotins, if they're if you're exposed during development, can change those cells from becoming bone, and instead they become a fat lineage. And if that's not stressful enough, we know that organotins also influence the adrenal gland, which is, as I mentioned earlier, where stress hormones are created. And so they lead to higher stress hormones. They also are known to affect other parts of the brain, the hypothalamus and pituitary axis. And this is the master regulator of our entire endocrine system, as well as our ovaries and testes, leading to changes in fertility and sexual development in males. And so this is overwhelming. What I want you to just focus on here is that a single chemical can alter many different organs. And now I hate to tell you that these synthetic chemicals are everywhere, and there's lots and lots of them. And so we take medicines, and those medicines, in this case, I'm showing you birth control because it's an endocrine active medicine, can go into our wastewater and affect the fish living in these areas and can actually change their sex. These chemicals are in our water because our waterways are polluted in some cases. And we intentionally spray many endocrine disrupting chemicals as pesticides on our food. They're in the air. And we actually impregnate most of our materials with things like flame retardants that seem like a good idea, except that these chemicals affect our physiology. Many of the items that we give to our children to play with and our babies to suck on and to chew on have chemicals that are known to disrupt the endocrine system in them. We actually make so much of this that we are forming mountainous islands in the ocean of these endocrine disrupting chemicals. And not surprisingly, we have these chemicals in our bodies. And so this little graph shows the percentage of Canadians in this study who were tested that had flame retardants in their bodies. And the gray shows the percentage that had these chemicals. And you can see that almost everybody had a variety of different flame retardant congeners in their bodies. So this brings me to our first story today, and it's about pesticides and how they affect reproductive development. And this is a story about my dissertation work that I did in South Florida. And I went to 5 different locations, and my aim was to understand how agricultural land use and pesticides affected reproductive system of wildlife. And the wild creature that I decided to focus on was the giant toad. And I did this for a couple of reasons, but one was that amphibians are indicator species. And so they're sort of canaries in the coal mine, as they are very sensitive to their surroundings. And so if they get sick or are exposed to something and have health effects, then that tells us that other things might be affected soon. The other reason I picked these to work on is because their sex hormones cause males and females to look different. This is a female and this is the male. And so you can actually just look at them and see if their sex hormones are functioning properly. So at the agricultural sites around Okeechobee, the males don't look like this. Male toads at the agricultural sites look like females. And if we dissect them and look at their gonads, females have ovaries with yolked eggs, and males at the non-agricultural sites have testes that produce sperm. But at the agricultural site, some of the animals look like this, where they have both testes and ovaries. And this is not normal in amphibians. Amphibians are like us, where we typically develop either ovaries or testes. And when we look at the data, what you can see is that the percentage of individuals with testes and ovaries together increases with the percentage of agriculture in the area. The severity and the number of gonadal abnormalities also increased with agricultural intensity. And at the most agricultural sites, males also had decreased sperm production and testosterone. I want to take a second just to say that testosterone is testosterone. So it's a chemical compound, and I have a picture of, its chemical structure here. So testosterone in the toads is the same chemical that is in people. So it's kind of like the water that's in my body is the same chemical as water in somebody else's body. So water is water and testosterone is testosterone. It's the same chemical. And so where does that take us? I think it takes us to the question of what is happening to the people. And this brings us to our second story. And so what do we know? We know that endocrine disrupting chemicals alter human reproductive health. And we know that embryonic exposure to endocrine disrupting chemicals correlates with the incidence of birth defects in humans. So babies that have high levels of endocrine disrupting chemicals in their bodies are more likely to have birth defects. So one of the things that my lab studies is how endocrine disrupting chemicals affect development. But knowing the mechanisms through which chemicals induce harm doesn't stop that harm. And we can't ban all the toxic chemicals in the world. And we certainly can't ban them in the amount of time that we would need to stop human health effects as quickly as we would like to. So I argue that we need a new perspective. And I'm not saying we shouldn't ban toxic chemicals. I'm saying that, that we should be responsible, and where chemicals are harming humans, we need to come up with alternatives. But there are so many chemicals in our environment, it's unreasonable to think that we're just going to stop using all of these chemicals. And so what we need is an alternative approach. And I believe the beginning of that alternative approach begins by changing the question that we typically ask. So in toxicology, we usually ask and try to figure out what causes toxicity. So when I was telling you about the organotins, I said, we know that these effects happen because these mechanisms have been worked out. These very specific things are known to cause these effects. And that's all good stuff, and we need to know that. But I believe that we can make progress if we change the question a little bit. And some of us focus on a new question, which is, how do we become resilient to pollutants? And so I can show you a little picture. In toxicology, we do these dose response curves, right? So we have this toxicant dose, and we expose different individuals, usually animal models. So these are each a different individual exposed to a certain level of this toxicant. And then we look at the response. And in this case, because this talk is probably stressing you out, we'll just say that the response is stress. And so with increasing doses of this toxicant, we get increasing stress. Now, typically in toxicology, we focus on the mean effect. So we don't focus on these individuals that have the strongest effects, because that might overestimate the effects of the chemical. And we don't focus on these individuals that have the least effect, because that might underestimate what's happening to the population. And so we focus on the mean. But I've come to become interested in these individuals that show a lower response. And so what's interesting about these individuals is that they have something that's making them resilient to the toxin. So why are they resilient? Maybe they have antioxidant enzymes or detoxification enzymes that the other individuals don't have. And so if we increase those, if we can figure out what they are and then boost them, can we increase resilience and bring down the response of everybody in the population? So this brings me to this idea of studying mechanisms of resilience. And so one context within which we can study this is fetal development. And so we could think about developing a prenatal supplement that we could use to reduce the effects of toxicants and protect babies during development. And to test this idea, we need to be able to reliably induce a birth defect so that we can test whether or not supplements can decrease that birth defect. So that means we're not going to be doing this in humans. We need a model system, an animal model. We also need to be able to quantify the severity of this birth defect so that we can ask, when we give this supplement, do we decrease severity? And we need a protective supplement. And so over the past 8 or 9 years, I have developed the system within which we can ask questions about protecting developing fetuses from contaminants. And so we have developed a system within which we can cause hypospadias 100% of the time. So we know exactly when to expose the pregnant mom to vinclozolin, which is a pesticide that is sprayed on a bunch of different vegetables. It's used mostly outside of the United States, but we know exactly how much vinclozolin to give to cause hypospadias 100% of the time. And so if we see no hypospadias, we know that we've changed that with that prenatal supplement. We also have developed a standardized scoring system so that we can score the severity of hypospadias. And we've found a protective supplement called sulforaphane that upregulates detoxification and antioxidant enzymes. I just want to take a second to talk about this supplement. So here on the y-axis, we have anogenital distance. This is the distance between the genitalia and the anus, and it is a measure of masculinity. It is correlated with testosterone function during development. And what we see is that when we increase sulforaphane dose, we can increase this anogenital distance. Now, it's important for you to understand that all of these individuals on this graph have been exposed to a level of vinclozolin that causes 100% hypospadias. So it reduces masculinity. It reduces the function of testosterone. And what we can see is when we have no sulforaphane, anogenital distance is low. And as we increase sulforaphane, masculinity increases. And if somebody is interested in this end of the curve, I'm going to let you ask that question at the end. So the idea is that we can increase these antioxidant and detoxification enzymes with sulforaphane and reduce the effects of vinclozolin. And so what I'm showing you here is hypospadias incidence, so the proportion of individuals within a litter that had hypospadias. And this top line is when there was no sulforaphane added, and this bottom line is when sulforaphane was present. And so we are able to decrease the incidence of hypospadias, even at these very high doses that lead to 100% hypospadias. And we also can decrease severity. And so by adding sulforaphane, we reduce the way in which the severity of vinclozoline's effects. So that's great. That was really exciting data. And where it leaves us is with this question of how does it work? What are the mechanisms? And so we know that sulforaphane increases antioxidant and detoxifying enzymes. And before I go any further, I just want to show you what a typical cell looks like. A cell has a cell membrane, and on the inside, what is colored blue here is the cytoplasm. And it's filled with these other organelles, and it's also filled with proteins that do things, that make things happen in the cells. And there's this nucleus. So what we're going to be talking about is the cytoplasm, the proteins in the cytoplasm, and this nucleus. The nucleus is where the DNA is. And so in the case where there's no sulforaphane, out here in the cytosol or cytoplasm, there are these proteins that bind to this protein called NRF2. And when there is no sulforaphane, these col3 and keep1 proteins target NRF2 for degradation, to be destroyed. And it blocks the ability of NRF2 to go into the nucleus. But when we add sulforaphane, it kicks this NRF2 off of col3 and keep1 and allows it to cross into the nucleus, and it decreases the destruction of NRF2, the protein NRF2. So what is this NRF2 doing? When it goes into the nucleus, it binds to the DNA at very specific genes and causes those genes to become transcribed. So the genes are transcribed, they make mRNA. mRNA can be converted to proteins. And the mRNA that is made are mRNA that are for antioxidant and detoxification genes. And then through, once they leave the nucleus and go out into the cytoplasm, they are made into proteins. And so we wanted to know if that rescue effect that we found was caused by this NRF2 protein. And so the way that we did that was to expose or to mate male and female mice that were heterozygous for NRF2. So the wild types these males had one copy of NRF2, one chromosome had NRF2, and one chromosome does not have NRF2. And when they're mated with females that have the same genotype, the females will carry babies that are either wild type or knockout or heterozygotes. We exposed these pregnant females to either our corneal control, sulforaphane, vinclozolin, or the combination of sulforaphane and vinclozolin. And we wanted to know if the animals that have no NRF2 still show that rescue. If they do, then we know that NRF2 is not required. And so we're barking up the wrong tree with the mechanism of rescue. And so again, I'm showing you anogenital distance. And what we found was that corneal individuals, the control, have a large anogenital distance. Individuals given sulforaphane also do. Those exposed to vinclozolin have a low anogenital distance. We knew that was going to happen. And what we find is only the wild type individuals with NRF2 show an increase in anogenital distance when we give sulforaphane with the vinclozolin. So only those individuals with NRF2 responded. And we see very similar response with hypospadias severity. Corn oil and sulforaphane have very low scores for hypospadias severity. Vinclozolin induces hypospadias and increases severity. And only those individuals, those wild type individuals with NRF2 had reduced hypospadias severity when we gave sulforaphane with that vinclozolin. So we know that the rescue requires NRF2. And we can protect urogenital development with sulforaphane. But if sulforaphane is broadly protective, then its effects must occur outside of the genitalia. So if sulforaphane is going to protect us from more than just this one chemical that targets the genitalia, it needs to affect organs outside of that target tissue. And so one target organ for sulforaphane to be working through could be mom's liver, but there are multiple other organs to consider. And because we know that brothers that were knockout and wild type within the same mom have different responses to the sulforaphane, we know that it can't be mom that's driving these effects, because the mom, if her liver was increasing protective enzymes, she would protect both of her babies. And so although the mom is interesting and we're studying her liver, the effects that are caused by sulforaphane that lead to this correction and reduction in hypospadias severity have to be occurring in the fetus or its placenta. And so we studied the placenta and the liver, and to investigate the physiological changes that cause the rescue effect, we ran proteomic and transcriptomic analyses on each of these organs. So the mom's liver, the placenta, and the baby's liver. And I just want to show you that transcriptomics is basically measuring the transcripts from the DNA. And proteomics allow us to look at what those transcripts are turned into. So what the proteins are. And I'm not going to go through all of that data because this is a story and not a dissertation, but I'll give you a summary. So what I've shown you is that sulforaphane decreases the potency of vinclozolin. We know that that rescue that sulforaphane causes requires NRF2. We have found that the fetal liver is not driving the rescue. It's probably being protected by sulforaphane rather than launching protective pathways. The placenta is broadly affected. There's a few really cool things that we've found. One is that sulforaphane drastically reduces the vinclozolin-induced gene expression. So if we compare the control, the corn oil, and the vinclozolin animals, all these genes are different in the vinclozolin animals. And when we give sulforaphane, 80% of those genes are no longer different. And when we look at biomarkers of vinclozolin exposure, they're completely normalized by sulforaphane. So it reduces those biomarkers. And the mechanisms of resilience are diverse, but they involve changes in oxidative phosphorylation, cell signaling, development, and also the immune system. So my lab is taking this novel approach to studying toxins, and we're interested in trying to understand the mechanisms of resilience. So here where do we go from here? And I think, the next part of our story is about the challenges and studying, trying to figure out how we can move this information into humans, and trying to understand whether or not we can actually protect humans with sulforaphane or some kind of supplement that is similar. And so the first question is it safe? And so you can buy many different supplements from Amazon that have sulforaphane in them. And if being able to buy them on Amazon doesn't make you comfortable entirely, that's okay, because there are currently 81 studies that are investigating sulforaphane, and all of them are investigating its safety, but in different contexts. And so, for example, this study is looking at how sulforaphane can protect against cardiotoxicity of breast cancer patients that are on chemotherapy that's very hard on the heart. People are studying it in the context of Parkinson's disease and schizophrenia, and also non-alcoholic fatty liver disease. So sulforaphane has been studied for many years and is being intensely studied. And so the idea that it could be used in humans is not a new idea. It's being investigated scientifically for use in humans, and people are using it. I actually take sulforaphane. So that brings us to what I'm doing at HBOI and the sort of story that's happening right now. And so one of the things that I'm focused on is harmful algal blooms. And these harmful algal blooms produce toxins, and Lake Okeechobee is one of the places that is affected. And because Okeechobee is affected, the downstream areas are also affected. And so one thing to know is that these harmful algal bloom toxins pretty much affect everything. So they affect, the nervous system, the lungs, the liver, the kidneys, and our gut. And so that makes it really hard to diagnose because everything is affected. We also know that sulforaphane protects humans from air pollution. And so there are several studies that have looked at how well sulforaphane can protect people from a number of different air pollutants. And it's safe and effective. And it enhances detoxification. Another thing that we know about sulforaphane is that in mice, it prevents microcystin-induced oxidative damage and apoptosis, which is cell death. And so microcystin is one of the more common toxins in blue-green algae blooms. And so this raises the possibility that similar protective effects could happen in human populations. So sulforaphane might protect humans from microcystins. So this brings us to this question: can sulforaphane help communities around Okeechobee and those surrounding Okeechobee? And we know that it's not just HAB toxins that the communities are exposed to. They're also exposed to agricultural pesticides, as well as air pollution. So ultimately, we would like to measure toxin levels in the humans living around Okeechobee and surrounding areas, and then provide supplements that have sulforaphane in them, and ask how those toxic levels change, and ask how health parameters become better. However, before we can test hypotheses about the effects of toxins on humans or investigate how we can mitigate those effects, we need to build trusting relationships with stakeholders to form the foundation of recruitable participants. So we need people to want to be involved with this. And so you can imagine your picture here. So we just recently received funding through the Harbor Branch Oceanographic Institute Foundation. And this money is aimed at the Florida Center for Coastal and Human Health. And I'm a member of this center. And through that center, I'm working with 2 collaborators to begin to form this foundation of participants that will be interested in being involved with the research to ask if sulforaphane can reduce toxic effects. And so Dr. Barbarite and Dr. Harris are both collaborating with me, and together we will do three things. Where this work will allow us to conduct outreach to educate our community about how human-induced environmental harm affects human health. So if we harm the environment, that harm comes back to us. And I think that link is a little bit difficult sometimes for people to see, but it's really obvious when you think about toxins. You know, we're putting those out there and then we're exposed to them. We want to also determine the concentration of HAB toxins and other pollutants in aquatic systems that humans visit or live near. And we also want to, through these outreach and investigations of water quality, build a network of community members that are empowered to become involved in this HAB research and in clinical trials. So I'm going to end things there so that we have plenty of time for questions. And before I go, I want to acknowledge that there were lots and lots of people that helped with this work, and a lot of funding that went into it. And I want to point out that the HBOI Foundation is currently funding a lot of the work that we're doing now. And with that, I think we have plenty of time for questions.
Great. Thank you very much, Krista. It's a little bit disconcerting, especially when you started out talking about all these different toxins where, just one of them can have a tremendous effect on our bodies. And you mentioned that, maybe the best way to deal with it is not necessarily trying to prevent it because they're everywhere and in our environment, but to try and counteract them. But it seems like, at least to some extent, or for some of these toxins, it might be the easiest to prevent them. Do you have any recommendations if you wanted to prevent any behaviors that you would recommend to prevent at least some exposure?
Sure. There's actually a lot of things that we can do, both to help our health and to help the health of the environment. And so there are lots of things that we can do that do both things. So for example, eating fresh food is really a good choice. And so some of us don't have that choice. Some people don't have access to fresh food. But fresh food is good for 2 reasons. One is that it has vitamins. It has, the precursors of sulforaphane, in broccoli and leafy green vegetables. Those antioxidants, in carrots and in peppers are really important and protective. The other thing is that they don't have preservatives. So wash them. I wash my vegetables with soap and I rinse them. If I wash my dishes with soap, I'm going to wash my food with soap. And so you have to wash your vegetables very well. There is a list through Environmental Working Group that every April, I think, they give information on the Dirty Dozen. And there they say, of all of the things that you buy organic, you can buy these 12 things organic and everything else don't. If you don't want, so there's different reasons to buy organic, right? I buy organic and I try to buy everything organic that I can possibly afford because I'm supporting the industry, the organic industry. But, as an undergrad, I couldn't do that. And I didn't have the money. And so if you just buy those 12 vegetables, then you will protect yourself. And they say that it reduces your exposure by 80%. So that's one thing that you can do. The other thing is to not eat a lot of canned food. Canned food cans have lining in them so that you don't taste the can. And those linings are endocrine disrupting chemicals. And so the information for how to protect yourself from these chemicals is out there. And I would be happy to sort of search through a couple of my favorite websites and give them, to you so that you can post them.
Yeah, so maybe we can post it on our website together with a recording of today's presentation. So we'll do that. Just as a reminder, if any of our audience have any questions, please put them in the Q&A button at the bottom of your screen. I see that questions come in, so I'm going to start going through them. So there are a lot of questions related to the harmful algal bloom here in Florida. And is it that Florida has the most toxic waters, or are other states affected, other regions affected from harmful algal bloom?
Unfortunately, harmful algal blooms are quite common really all over the world. Florida has a lot of water that we care a lot about because of tourist reasons, but also because they are we depend on those waters for our own livelihoods. And so at HBOI, we're focused on our local area. And I believe that work that we do here we will be able to generalize to other areas. But the Great Lakes are affected. There's a lot of algal blooms in the Northeast. So, and yeah, other places on Earth.
Yeah. You mentioned the toxins that are associated with harmful algal bloom that are derived from there. The question here is whether these are toxic just when drinking the water, or is it okay to swim in it? Is there a difference, how the toxins are basically absorbed from the body, by the body?
So it turns out that our skin is a really great barrier. But we can't, so the problem really is with the blue-green algae, the problem is getting them into your body through drinking contaminated water, or if you were out swimming, drinking the water that has the harmful algae in it. So, that's the problem. One really terrible problem is that dogs that go swimming get the algae on them, and then they go and clean themselves and can die. And so actually, one of the things that I want to study is whether or not sulforaphane supplements that they make for dogs can be used to protect our dogs from harmful algal blooms.
Interesting. Now, just one more questions related to the toxins in the water. Several houses and several house owners put water filters in. You can have this Brita filters, where you filter your water. Will that get rid of these type of toxins?
Yeah, really the best way to filter your water is through carbon. So a lot of those things have carbon filters. Carbon and the very fine mesh filters. So yeah, that's helpful.
Yeah, good. There are a couple of questions related to sulforaphane as a supplement here. So one is, what is the suggested dosage of sulforaphane? You mentioned you're taking it yourself, so maybe you can speak just from your own experience.
So, that's funny that I don't know the answer to that since I take it. I need to look back at the bottle, but it's just once a day. And it's, the sulforaphane, that the amount of sulforaphane that you would have to, the amount of broccoli you would have to eat in order to get the dose of sulforaphane that we're using in these studies is like too much. Nobody wants that much broccoli. And so it's nice to have this, in a pill form where it's concentrated. And so there are a variety of different supplements that have, different concentrations. And so I would have to look across those different supplements to see, what the concentration is. One thing I will say is that I've kind of been a little lax in my usage of the word. So in our studies, we use sulforaphane that we get from a company that synthesizes the actual sulforaphane chemical. A lot of the supplements use precursors to sulforaphane that when you take them, your body makes the sulforaphane internally. And so the doses are a little bit hard to compare from my work to the actual supplements. I don't know if that answers the question. If not, I'm happy to keep going.
Yeah, and feel free to reach out to Dr. McCoy and directly and chat with her about specifics to the sulforaphane and the origins of it. It's curious because the cruciferous vegetables, especially broccoli, has also been implicated to have anti-cancerous effects. And again, the amount of broccoli you'd have to eat to actually see that effect is tremendous. Do you see any correlation between the effect it has on these toxins and the anti-cancer effect that we're seeing with the same type of vegetables?
Yeah, I mean, there are several studies looking at the effects of sulforaphane to reduce development of cancer. That's, if you look through those clinical trials, there are a lot of studies that are investigating how sulforaphane can protect you from developing cancer. And so one of the ways that might be happening is by decreasing oxidative stress. And so, and therefore protecting the DNA so that it doesn't mutate and doesn't become cancerous. And then in another context of cancer, they are studying sulforaphane as a way to protect people that are on chemotherapy. So it decreases the toxic effects of chemotherapy. And I'm just going to throw this out there. There are a couple of studies in very specific cancers where the cancer cells will upregulate NRF2 to protect themselves. So it's complex, of course.
Of course, definitely cancer is a complex disease for sure. So we have a participant here who does a similar study to your harmful algal bloom study on Clear Lake. And the question here is how you're going to correlate the aquatic cyanotoxins with the health impacts, whether you're just going to measure toxins in ambient water or if you're also going to look at fish tissue, drinking water, inhalation, those kind of measurements, and then correlate them.
I should have, that reminds me of another study that we're thinking about that I didn't present. So the question of exposure is a hard one because there are so many different ways. If you're just thinking about microcystin LR, this one version of microcystin that is found in blue-green algae. There's just that is, there are many different ways to be exposed. So through the air, through the food, through the water. And then you need to think about the fact that we are not only exposed to microcystin LR or just microcystins or even just toxins from blue-green algae. There are other algae, right? I've just mentioned blue-green because, I was going to say grew up. I didn't grow up in Okeechobee, but I feel like I did. I did my dissertation there. I spent a long time there. And so that's sort of what I'm thinking about now, is like getting back to Okeechobee and helping the people rather than, well, not rather than, but in addition to helping the frogs, right? And so for me, what I'm interested in is measuring what's in the people. So what is the exposome? So how many different chemicals do I have in my body? And can we get rid of them? And so I don't want to say I don't care how they got in there, because as you mentioned, if we know that, then we can change our behavior to help prevent getting them. So if I know that it's in my water, well, I'm going to stop drinking that water, or I'm going to filter that water. If I know pesticides are on my food, I'm going to clean my vegetables before I eat them. So I guess the way that I'm thinking about this is really to measure what's in our body and what can we do about it.
But then you still have the difficulty not knowing where it's actually coming from, right? To really bring it back to, okay, the source of this chemical is the harmful algal bloom, or whatever you're studying. So as you mentioned earlier, it's very complex.
Yeah. And I'll just say that where these things are coming from are various, and that's a great question for environmental toxicologists. That isn't necessarily what I'm focused on answering, but it's important and should be done, because if we can identify where these things are coming from, then we can change our behavior. But that's not necessarily what my focus is.
Mm-hmm. Completely different question. You mentioned earlier that there's a need for organism model systems. Of course, we cannot just go out and test things on humans, and we don't want to do that. You mentioned the study earlier where you had toads. How closely related are results in toads to those found in humans?
Well, there are many similarities between reproductive systems of animals. I could talk about really interesting sort of oddities in the animal kingdom, and like how reproduction is, it's just fascinating, and it can be very diverse. But in general, the way in which our reproductive systems work is conserved. And so, the point that I made about testosterone, that's the same chemical in the frogs, and it's produced through the same pathway, the steroidogenic pathway, in many different organisms. And so if we have chemicals that are altering the development or the synthesis of those hormones, they're affecting the synthesis of those hormones. And so I expect people that are living around those frogs to have endocrine problems with their reproductive system. I have not measured that. I would love to, and I would love to see if we can clean that up and change it with sulforaphane, or some other, I've just focused on sulforaphane because it works, in my models. But it could be any number of things that sort of, I think, turn on NRF2, that master regulator of those protective genes and proteins.
Okay, we're almost out of time. I will finish us off with one question, which is actually relatively scientific. I apologize to those who still have questions. We will ask Dr. McCoy to answer those offline. We will post those answers on our website. So you do get an answer to your questions. Okay, so the last question before we have to go, does hypospadias also result in retained ovaries and testicles, or is it only a urethra abnormality?
So hypospadias occurs when the urethra does not form in the proper place. So it's not at the distal tip of the penis, but somewhere along the shaft or the perineum. That's the definition of hypospadias. Now, what the mechanisms that cause that birth defect are not only causing that one birth defect in many cases. And so what happens with vinclozolin in particular is that it literally binds to the androgen receptor. So this is the protein that testosterone has to bind with in order to work. And so vinclozolin blocks that. And so testosterone can't work. And so you don't get masculinization occurring properly. So hypospadias can occur with testes that do not descend properly. It is in many cases, if you have hypospadias, you are at a higher risk of testicular cancer. You're at a higher risk of multiple reproductive abnormalities, because it's not like the urethra is forming in isolation. And that's the whole thing about these chemicals that affect the endocrine system. The endocrine system is basically the chemical plumbing rather than the blood vessels. It's the chemical signals that drive development. And so when you start sort of plucking those out and changing those, it changes the whole system. It can.
Yeah.
And it depends on the dose of the toxin. It depends on when the individual is exposed. It depends on, probably mom's background, detoxifying enzymes. So our genetics.
Very complex, definitely. Okay, we will have Dr. McCoy answer the remaining questions offline. We will post those answers together with a recording of today's presentation on our website. Krista, thank you very much. A very interesting presentation, very important. We hope to see everybody back next week. Have a great rest of your day. Bye-bye.
Bye.